Hello, and thank you for joining me as I demonstrate how to analyze the results of a simulated genetic cross using FlyLab JS. In this video, I will show you how to use FlyLab JS to conduct a simulated genetic cross, how to interpret the results of the cross at each generation, and how to use the data to test the results using a chi-square test. Imagine you're part of a genetics research lab, and you've just successfully completed your first genetic experiment. You have some data that you need to analyze to determine if the trait is inherited according to Mendelian principles. In a previous lesson, we designed a mating pair. The female was wild type with red eyes, and the male fly had purple eyes. To begin the experiment and mate the parent, select the Mate Flies button between the two flies. The result of this cross is the first population of offspring, which is called the F1 generation. We see the parents at the top of this screen. At the bottom of the screen is a description of the F1 generation. Here we see that all the offspring have the red eyes like their mom. From these results, we can determine that the red-eyed trait is the dominant trait and the purple color is recessive. To get to the next generation, called the F2 generation, we will select the mating pair from the F1 generation. This is done by clicking the Select to Mate button under the female and male fly from the offspring of cross number 1. When that is done, we're back on the Mate tab, and we will click the Mate Flies button. This will give us the results of the second cross, the F2 generation. What we see is there are female and male offspring with either the wild-type red eyes or the recessive purple eyes. We can also see how many of each type of offspring have been produced. There are 368 red-eyed females, 407 red-eyed males, 135 purple-eyed females, and 125 purple-eyed males. With the F2 generation completed, we can begin to test the hypothesis. So we will now select the Analyze tab at the top of the page. Doing this lets us see the results organized in a data table. We see the same data as before. Notice the numbers are the same. The plus sign indicates wild type or red eyes, and the PR indicates purple-eyed offspring. For this analysis, we're not interested in the proportions column. To simplify the analysis, we're going to select the Ignore Sex of Flies box above the data table. This will combine the data for offspring with the same trait into one row. What we see now is a simplified data table showing that there are 775 total wild-type offspring symbolized by the plus sign, and 260 total purple-eyed offspring symbolized by the PR designation. To complete the test of the hypothesis, we will now select the Include Test Hypothesis box above the data table. We see that more columns have been added, including hypothesis, probability, expected, and a calculation column. The only column we need to deal with is the hypothesis column. The other columns will be filled in later. Because in the cross we made, we kept track of only one trait, eye color. This was a monohybrid cross, and according to Mendelian genetic principles, the expected ratio is 3 red-eyed flies for every 1 with purple eyes. So in the hypothesis column, we're going to put a 3 in the first window and a 1 in the second window. We are now ready to test the hypothesis. We do this by clicking the Test Your Hypothesis button at the bottom right corner of the data table. The program has now filled in the remaining columns with the values based on the hypothesis we submitted. To interpret the calculations and make a conclusion about this cross, we're going to refer to the level of significance line below the table. In this case, the level of significance is 0.9285. Since the results are so close to 1, the level of significance is considered high. Because of this, we can conclude that the eye color trait is being inherited according to Mendelian principles. Great job! You have now successfully completed a genetic cross, analyzed the results, and made a conclusion about the inheritance of the trait. You are becoming a valuable member of this research team.